Hello, my name is Dana Cavadon, and today we are going to be talking about the anatomy and enzymology of fat absorption. So here I have a picture of the overall digestive system, all of the organs a part of the digestive tract. It starts in the oral cavity, also known as the mouth, and it moves on to the GI tract, which includes the esophagus, into the stomach, um, from the stomach into the small intestine, and then the large intestine, and then feces are secreted out of the rectum to the anus. So we're first just going to talk about fats. Fats include triglycerides, cholesterol, phospholipids, long-chain fatty acids, and fat-soluble vitamins. The most common form or type of fat found in the human body is they're found typically as triglycerides, and we have a picture of that seen here. And then we also do see a large amount of cholesterol in the human body as well. Lipids are not water soluble, they are hydrophobic. This also means that they are lipid soluble and lipidphilic. So that means that they can diffuse or travel through lipid membranes. We will get a little bit more into this about fats being hydrophobic and lipid soluble when we talk further about the actual fat absorption through the epithelium of the small intestine. One thing I wanted to show here quick was just the triglyceride is made of a glycerol and 3 fatty acids. So going back a little bit to the digestive system, with this picture in the bottom left, food that is high in fat content, first of all, gets chewed in the mouth and then is swallowed. It makes its way down the esophagus and into the stomach. There's a little bit of fat digestion in the stomach with the help of gastric lipase, but we really want to focus on what leaves the stomach, and that is chyme, and chyme is very aqueous. So since we know that fat is hydrophobic and the chyme is very aqueous, it needs to be encased in something in order to be transported with this chyme into the small intestine in order to be absorbed. So this is done through the use of bile salts, and bile salts come from bile, which is secreted by the liver. Bile is a non-enzymatic solution that is secreted from hepatocytes or liver cells through the hepatic ducts. You can see in the picture in the image in the bottom left of the slide here that coming down from the liver and combining with the cystic duct is the common hepatic duct that the bile is secreted through. Yep, so in the liver, the hepatocytes, there are small channels in which bile is secreted. Those are called the bile canaliculi, and they, multiples of those, they coalesce into bile ductlets, which run through the liver alongside the portal vein that you can also see in the image here. So the common hepatic duct takes bile into the gallbladder for storage, and then the common bile duct takes bile from the gallbladder into the lumen of the small intestine in order to aid in this fat absorption. So during a meal that includes high fat content, there's contraction of the gallbladder, which sends bile into the duodenum through the common bile duct, as I just mentioned. But the fatty acids in the intestine do this by stimulating the release of cholecystokinin, which is a hormone that targets the gallbladder and the pancreas. So it causes that gallbladder contraction to release the bile, and it also causes the pancreas to secrete enzymes. And we'll get to that in just a minute. But some key components of bile. Bile includes bile salts, bile pigments, and cholesterol. Bile salts help felicitate the enzymatic fat digestion. Bile pigments are waste products from hemoglobin degradation. And then again, just cholesterol in the human body. So bile salts act as detergents to make fat soluble during digestion. Bile salts are hydrophatic, which means they both hydrophilic and hydrophobic regions. So the hydrophobic region is obviously the region that is facing in towards the surface of the lipid or the fat. And the hydrophobic portion is the one that interacts with the water outside of this molecule. So this helps create a stable emulsion of small water-soluble fat droplets. So that's how the fat is in the chyme in the small intestine. So the aqueous chyme leaves the stomach, and that contains large emulsions, and these bile salts help break down these emulsions into smaller, more stable particles. So enzymatic fat digestion is carried out by an enzyme called lipase, and lipase is secreted from the pancreas. It's secreted specifically from acinar cells. So the exocrine portion of the pancreas is what consists of lobules, and those are called acini. And it's from those acinar cells that lipase and colipase are secreted. And it's the ducts from these acini cells that empty specifically into the duodenum of the small intestine. And that's how these enzymes get to the fat in the small intestine. So as lipase, as an enzyme, it removes 2 fatty acids from each of the triglyceride molecules. So that results in 1 monoglyceride and 2 free fatty acids. The unfortunate thing is that lipase is unable to penetrate the bile salts itself. So fat digestion also requires the enzyme colipase, and colipase is a protein cofactor that is also secreted by the pancreas, as we just mentioned. Colipase displaces some of the bile salt so that lipase can have access to the fat or the lipid inside the bile salt coating. So lipase and colipase digest the triglycerides, and again, that results in 1 monoglyceride and 2 free fatty acids. Those 2 free fatty acids and monoglyceride are absorbed by simple diffusion across the apical membrane of the small intestine. Then they move out of what are called micelles, which are small discs with the bile salt, some phospholipids, some other fatty acids, cholesterol. But they move out of the micelle and diffuse across the membrane, the apical membrane, and they are just doing simple diffusion. And then once they're inside the cell, they move to the smooth endoplasmic reticulum, where they actually recombine and reform into triglycerides. The triglycerides are then joined by cholesterol and some proteins to form a large droplet, and that's called a chylomicron. This chylomicron is then packaged into a secretory vesicle by the Golgi, and is then secreted out of the cell through exocytosis. And that's secreted across the basolateral membrane. Once out of the cell, the chylomicrons are actually absorbed into lacteals, and lacteals are part of the lymph system. And then once they're in the lymph system, that's how they get back to the blood system. They pass through the lymph system and enter the venous blood just before it flows into the vena, the right atrium, which is the vena cava. And then this is just kind of a picture of everything that is going on through what I just described, the fat absorption. So large fat droplets, for example, are triglycerides and cholesterol. They form with bile salts. The bile salts coat these fat droplets into large emulsions, and then the bile salts, again, make them smaller in order to be more stable and more easily digestible with larger surface areas. Then pancreatic lipase and colipase come in and break them down into monoglycerides and free fatty acids. These are then stored in the micelles, so the pancreatic lipase and the colipase breaks the micelles down into the monoglycerides and free fatty acids. Those then move out of the micelle and through the cell through simple diffusion through the apical membrane. Once inside the cell, the absorbed fats go to the smooth endoplasmic reticulum to reform into triglycerides. They are then added with cholesterol and proteins to form chylomicrons. The chylomicrons are absorbed into a secretory vesicle through the Golgi apparatus to be excreted out of the cell, and then from there they go into the lymph system to the vena cava.